All right, take it away, guys.
Awesome. Well, thank you, Beth, and thank you to all for joining us. We're very excited to talk with you all and about neon. So let's get started here. Jarrett's going to run the clicker, so I'm going to have to say next slide every time. So next slide, Jarrett. I've only got a few here. I'm going to try to keep my cats quiet. Okay, so just to give a quick overview of what we're going to talk about. I'm going to give a very brief overview about what the NEON project is and kind of overview of our data products. And then I'm going to turn it over to Jarrett to talk about what you're all really here to talk about, or hear, the Flora protocols for about 30 minutes. And then we've got some cool Flora data visualizations and case studies that Jarrett's going to kind of talk about for 15 minutes. And then should have 10-ish minutes at the end to go over some questions. So let's start with that overview. So, oh, no, you're good, Jarrett. Yep. Okay, perfect. So what is NEON? So the National Ecological Observatory Network is a program that's sponsored by the NSF and operated under a cooperative agreement by Battelle Memorial Institute. So the material in this is based in part upon work supported by the NSF through the NEON program. So a lot of words there, but the NEON is basically a 30-year project, and we provide free and open data on the drivers and responses to ecological change, standardized, reliable framework for research and experiments. And really, we want to provide a highly coordinated national system for monitoring a number of crucial ecological and environmental properties at multiple spatial and temporal scales. So kind of a lot to unpack there, but if you go to the next slide, Jarrett, what we really want, the intention of NEON, what we really want to do is we really want to take that reliable standardized framework and that really solid foundation of data to enable understanding and forecasting of the impacts of climate change, land use change, and invasive species on continental scale ecology. So that's really what we want to provide, is that stable framework for other people to forecast and understand ecological change. So continental scale ecology, what does that mean? Next slide, Jarrett. So that means basically everything except for Mexico and Canada. So we have offices on the North Slope of Alaska. We have field sites, and we have field sites in Puerto Rico and one in Hawaii and Massachusetts and Texas. So we're everywhere. So we have 81 field sites. We have 47 terrestrial, 34 aquatic sites. We'll get into a little bit later. And we're generating over 180 different data products from those sites. We're not going to talk about all the data products today, but we will get into a few of them. So yeah, next slide here, Jarrett. So let's zoom in on domain 12, which is well, Yellowstone National Park. That's where we work. So here's a map of our site. It's a little small, or a little fine detail. I apologize, we can't see it too well, but you can see on the left there is basically Undine Falls, Jarrett, if you want to laser point that. And then on the right, far right, is Tower Junction. So our field site, yeah, and you can see the grayed in shaded area. That's our terrestrial sampling area. So basically we have plots all throughout there, and those little green and red dots are our plots where we take samples from. And so, yeah, so we're in the northern range of Yellowstone National Park. So we basically follow the loop road, more or less, from Undine to Tower Junction. In that blue box there, you can see zoomed in on Blacktail Creek, which is our aquatic site. So we have the terrestrial site and then the aquatic site. And all those dots on that zoomed in map, so that's where our aquatic infrastructure is placed, and that's where we take our aquatic samples from. And then the green box zoomed in is our tower area, where the NEON tower is, and the blue dots in the green square are our tower plots, which Jarrett's going to talk a little bit about later, but different types of terrestrial plots. So that's sort of a broad overview of NEON and Yellowstone. So I think we can go to next slide. So how does NEON work? What do we collect here? So let's give a brief overview of that. So next slide, Jarrett. So you can fit NEON's data collection methods into three big buckets, sort of. So the automated instruments that are in situ or at the site constantly collecting data. There's the observational sampling, which is what Jarrett's going to talk about. And then there's the airborne observation or the airborne remote sensing platform. So that's airborne remote sensing is a different department within NEON, so we don't interact with that too much, but I will give a brief overview because it's really cool. Basically, we have these airplanes that are equipped with high resolution cameras and hyperspectrometers and LiDAR system, and they fly over our sites near peak greenness. We can get really, they can generate some really high quality like vegetation and topographical maps. I mean, it's really, really cool what they can do. But that's not what we're talking about today. So those are sort of the three main buckets of NEON protocols. But let's talk about those first two a little bit more in depth. So next slide, Jarrett. So the automated instruments, automated instrumentation systems, they complement our field observation sampling. So these are sensors that are, like I said, they're continuously just out in the field collecting data. So they collect water quality data, solar radiation, precipitation, weather data, while measuring carbon and water cycles and uptake through the system. So on the left there, that's our terrestrial site, or terrestrial instrumentation system, which is our tower. So that picture on the left is our tower. I don't know if any of you have been snowshoeing through Blacktail Plateau Drive and seen this big old honking piece of metal out there in the forest. That's ours. Yeah, it is a pretty huge thing. Lots of instruments on there, and it's an eddy covariance tower, which is basically getting carbon uptake measurements throughout the system. And then the aquatic system on the right there, that's, like I said, at Blacktail Creek. There's the mini, it's like a mini tower on the top left there, MET stations we call it. Then there's in-creek sensors, which are getting dissolved oxygen, pH, turbidity, continuously monitoring all of those things. So really cool, and they complement our field sampling. So next slide, Jarrett. And so to kind of introduce the observation sampling, I'll let Jarrett take it from here mostly, but just to introduce it. So there's the aquatic side, which we're not going to talk about today, but then there's the terrestrial side, which is the fauna and the flora. So Jarrett's going to talk about the flora, but introduce the fauna. We do sample for ground beetles, mosquitoes, ticks, small mammals, and breeding land birds. And so this combined with the flora really gives us a really holistic picture of the terrestrial sampling sites where we are. So with that, I think I'm just going to turn it over to Jarrett to talk about plant stuff. He's got some cool stuff for us. So it's all you, Jarrett.
All right, thanks, Jake. Yeah, really excited about this opportunity to get to speak to y'all today. But we can go ahead and jump into the meat of this presentation, which is the flora work we're doing in the park. So here we have a list of our flora research protocols that we conduct in Yellowstone and our other sites, which include plant diversity, plant phenology, litter fall and fine woody debris, herbaceous biomass production, leaf area index, vegetation structure, below ground biomass, coarse down wood, and canopy foliage chemistry. And on the left there, you can see which protocols are designated as being part of our plant biomass and productivity suite, as well as protocols that have biogeochemistry elements. So to start out here, let's see. We have plant diversity. So this helps us provide an understanding of changes in composition, distribution, and abundance of native and non-native plant species. It is comparable within and across NEON sites and other continental plant diversity efforts. It also helps us understand impacts of drivers of change on the diversity of plant species and functional role in ecological systems. So for sampling plant diversity, what we basically do is we identify all of the plants in 33 of our plots every year. 30 of those are distributed across our site, and 3 of them are in our tower airshed. So we observe species and variable percent cover for our 1 meter squared nested subplots, and then additional species not yet observed in our larger nested subplots. So our sampling effort alternates year to year, where we sample just the 1 square meter nested subplots on 1 year, and then the full plot the subsequent year. So there are 6 of those 1 square meter nested subplots. We also collect archive vouchers, approximately 20 per year, which in future years there will be replicates of the more common species. And we also collect genetic foliar tissue of our 3 most dominant species every 5 years. So here on the left, you can see a layout of our nested subplots. So the blue squares being our single square meter plots, where those variables and percent cover values are recorded. And then we would move into the 10 square meter nested subplot, followed by the 100 square meter, designated there by 31, 32, 40, and 41. And then we'd repeat that flow for the next subplot, moving from 31 to 32. You can also see what one of our quadrats or square meter frames looks like in that picture on the right, where we have 10 centimeter markings on those to assist us with percent cover estimates. Next is plant phenology, where so plant phenology measurements record seasonal progression of critical biological processes and timing of ecological events, things like initial growth, flowering, and senescence. These measurements track indicators of biotic responses to climate variability by recording and monitoring phenological stages in plant communities. This helps us understand impacts of changing forces such as temperature, timing and duration of pest infestation and disease outbreaks, water fluxes, nutrient budgets, carbon sequestration, and food availability. Our sampling method for this involves status-based monitoring of individuals for approximately 50 bouts per year. We are currently in phase 1 of this protocol, which involves doing observations on 90 selected individuals, 30 of each of our 3 most dominant species, which I'll get to in a moment. In 2022, we will begin phase 2 of plant phenology sampling, so kind of establishing selection of the species and individuals for that phase this year. It will be 100 individuals, 5 of each of 20 common species, and those will include those 3 species we're currently observing in phase 1. We also have a phenocam that is mounted on our instrument tower, which does continuous monitoring of 9 selected phase 1 individuals, 3 of each species, which is also validated by in situ observations. So on the left here, you can just see kind of a rough representation of the layout here of our tower airshed. The gray boxes just being tower plots. That large box on the bottom left being a representation of our primary phenology loop, where we have those currently 90 individuals selected that we monitor. So each side of that is 200 meters, and the individuals selected are within 10 meters of the square transect. And then the tower, that is that yellow triangle there, directly north of the tower in view of the phenocam is our phenocam plot, where we have those 9 individuals, 3 of each of our most dominant species selected there. And then to the right, you can see the species that we are monitoring, which include Douglas fir or Pseudotsuga menziesii, sulfur flower, or Eriogonum umbulatum, and big sagebrush, of course, Artemisia tridentata. So moving on to litter fall and fine woody debris. Quantifying production of litter fall and fine woody debris is required to estimate annual above ground net primary productivity at a plot, a site, and continental scales. It provides essential data for understanding vegetative carbon fluxes over time. So our method here, we collect litter from elevated mesh traps throughout the year, and our samples are then sorted into functional groups, things like leaves, needles, flowers, twigs, etc. And we also collect from ground traps once a year to account for those larger particles, so leaves and twigs that are more than ½ meter long. This protocol also has a biogeochemistry element that we conduct every 5 years. On that interval, we take a sample from our highest litter production bout during the year, and we use the leaf and needle functional groups from that sampling bout for chemical analysis. So carbon, nitrogen content, isotope ratios, lignin, as well as archive samples. So here on the left, you can see one of our ground traps delineated there, which are ½ meter by 3 meters. So an entire one of our sampling cells. Then we have an example of one of our elevated mesh traps and an example of some sorting of that litter in preparation for drying and weighing. And obviously we deal with a little bit of snow in our traps sometimes. This is actually a photo from one of our northeast domains though. Herbaceous biomass production. So the net primary productivity of this plant group can contribute significantly to total net primary productivity, even in forested ecosystems. Understanding long-term trends in herbaceous community structure and biomass is very important in grazed systems where these plants constitute a critical food source for wildlife and livestock. In addition, members of the herbaceous plant community can respond relatively rapidly to various global change drivers. So for our method for this protocol, as a standard practice for herbaceous biomass and productivity, this is assessed via clip harvest during peak biomass. This is followed by sorting clipped material into current year and previous year's growth in order to estimate annual net primary productivity for this plant growth form. Current year growth is sorted by species into additional categories based on plant functional traits, these being cool season versus warm season graminoids, leguminous versus non-leguminous forbs, etc. In order to engender cross-compatibility with existing research, we sort clipped biomass into functional categories that are broadly similar to those employed by the Global Nutrient Network Research Group. Co-located below ground biomass core sampling also occurs every 5 years. And we'll get to that protocol here in a little bit. So here on the left, you can see the arrangement of our sampling cells that we randomly sample for this protocol, with our exclusion zones in red there, which are just kind of a 1 meter buffer around the edge of the plot, as well as our nested subplots that are used for more sensitive sampling like plant diversity and vegetation structure. And you can see one of our clip strips delineated here in some of these pictures, which are 10 centimeters by 2 meters. And then we have another photo here of some technicians sorting some samples there. So leaf area index, or LAI, is a useful proxy variable for numerous other variables of ecological interest, including plant biomass, plant productivity, forage quality, carbon balance, ecosystem energy flux, plant density, and the heterogeneity of plant cover. LAI is also used widely as a key input variable to models that seek to predict ecological processes such as carbon cycling. Regional to continental scale estimates of LAI are typically derived from satellite data, but validation of satellite data with aircraft and ground collected data is relatively uncommon. By leveraging NEON's aircraft and ground collected estimates of LAI, it will be possible to develop ground validated estimates of LAI at the continental scale. For sampling LAI, we use digital hemispherical photography, or AKA fisheye lens. And we do this at 3 tower plots using the same points and plots throughout the growing season, sampling every 2 weeks. We take upward and downward facing photos, upward only if there is overstory present in those plots. Also, every 5 years, we also sample our distributed base plots one time during peak green linked to our flyover by the aerial observation platform. So in the diagram on the left there, you can see our repeated points that we sample at 2, 6, and 10 meters for every cardinal direction from our plot centroid, and some different positions in those photos that we use to take those photos. So shoulder height upward facing, and the one down on the bottom is an extended monopole downward facing photo. Vegetation structure. The measurement of vegetation structure and the mapping of freestanding woody stems within our terrestrial observation system plots enables calculation of per stem, per plot, and site level woody biomass and productivity. Within tower plots, woody biomass increment is often a dominant component of total above ground net primary productivity. Understanding changes in woody biomass stocks and fluxes is therefore an important prerequisite for predicting ecosystem carbon balance. Recording the types and stature of woody species also provides insight into habitat quality and ecosystem responses to environmental change. Vegetation structure data are also an important complement to data streams generated by our airborne observation platform, or AOP. When combined with AOP data, these ground collected data will validate LiDAR data used to map the structural complexity of vegetation and will enable mapping of plant biomass at the site scale. In conjunction with carbon flux data from our terrestrial instrumentation system, vegetation structure data will facilitate understanding how biomass and different plant growth forms contributes to ecosystem level carbon flux. So our method for vegetation structure, we generate data describing the spatial location, structure, volume, and biomass of the woody stem plant community, including trees, sapling, shrubs, lianas, and other growth forms. Data are collected with handheld tools in the field using methods with a long substantiated history in the forestry community. We measure tower plots annually and distributed plots every 5 years. So on the left here, you can see someone measuring DBH, which I'm sure some of y'all are familiar with, or diameter at breast height. And then the second photo is someone measuring either a sapling or shrub at DDH, or diameter at decimeter height. And then this third picture is just an example of how complicated this protocol can get. So just kind of a mess of lianas in this photo, but this kind of brings me back. Definitely looks like a couple of my plots down in Texas. Below ground biomass mentioned earlier. So below ground biomass represents a substantial component of the total plant biomass and plant carbon in terrestrial ecosystems. Yet below ground biomass stocks and turnover remain very poorly understood, both in space and in time. Developing a better understanding of below ground plant biomass, as well as how much of that biomass is produced and decomposed within a given year, is therefore crucial to improving our understanding of how terrestrial ecosystems respond to environmental changes. So our method for this protocol, we sample every 5 years, taking 2 cores or monoliths at random locations in tower plots, which are co-located with our herbaceous clip for that sampling year. Our samples are then sieved and fine roots are sorted into size categories. Root fragments that are less than 1 centimeter in length are accounted for in our dilution sampling or subsampling method. Our samples are then dried and weighed, and a subset of those are ground for chemical analysis, carbon, nitrogen, lignin, isotope ratios, as well as archive samples. So on the left there, diagram again of our sampling cells within a plot, but a broken down sampling cell image there just to the right of that, where you can see our clip strip, our 10 centimeter by 2 meter clip strip in the middle of that 0.5 meter by 3 meter sampling cell. And at the end of each end, the north and south end of that sampling cell is where the associated below ground biomass core or monolith can be taken. So on the right, you can see some of our sampling using our root washing station for sieving and removing mineral material. On the bottom center there, we have someone sorting some roots using one of these metal wire gauge measurement tools. And then on the top right, you can see an example of us doing our dilution sampling, in which, so the residual fraction, or what is remaining after all of the qualifying roots are removed, is then diluted and agitated, after which an aliquot or a specific sample volume is taken from that agitated sample, placed into one of these metal tins on the bottom right, which is then split into fine root fragments and organic matter, so 2 tins per sample, and then dried and weighed. Coarse down wood. Monitoring stocks of coarse down wood, or CDW, is important because CDW uniquely influences ecosystem function. In terms of ecosystem services, CDW provides habitat for wildlife, stabilizes soil, increases microenvironment heterogeneity, stores carbon and nutrients over decades, and can enhance seedling germination for trees and other plants. Because coarse down wood particles can persist in the environment from decades to centuries, these particles have afterlife effects on ecosystem function of similar magnitude to those of live trees. In addition, knowledge about the quantity and size distribution of CDW pieces at the landscape scale can be used to model the probability of fire occurrence and severity, because CDW can be an important fuel source. So there are 2 components to our sampling method for this protocol, including volume and bulk density sampling. For our volume sampling, we use a line intercept distance sampling method, or LIDS sampling method, in which we do log tallies on transects with probability proportional to volume. So the length of the transect increases for large volume logs. I have a little bit more about that in just a moment, but for bulk density sampling, we are just cutting cross-sectional discs and determining mass and volume. So when we do tally, we are observing taxon ID, decay class and characteristics, as well as dimensions. And then when we do the subsequent bulk density sampling, we are going out and collecting those cross-sectional discs from that entire range of species, decay classes, and size classes that were observed in the tally. So both of these sampling components occur on 5-year intervals, but not necessarily on the same year. So it might be difficult to see some of those numbers there, but in the figure on the top left, it is, so this is showing kind of how one of our transects works. So for this particular figure on the top, the F equals 15, that is a volume factor that is assigned. And a volume factor can be different for each site, and that is determined based on vegetation structure data. So what is being shown here is on the bottom we have our round diameter equivalent for the logs that we're measuring, and on the y-axis on the left our limiting distance. So the distance at which that particular diameter will no longer be measured. So for example, a round diameter equivalent log of 50 centimeters is not measured beyond 70 meters there. Let's see if I can actually get my pointer there. Yeah, so 50 centimeter diameter log. You wouldn't continue to measure logs of that size, and they would need to be much larger the farther you go along that transect. So it makes it a little more efficient sampling effort. On the bottom here, you can see how we can encounter some more complicated logs that may be of different size classes or may be forked individuals. And then we have a diagram of our plot with our randomized 3 transects that are equidistant. And then just us running a tape there for a transect, and then cutting some cross-sectional discs with a chainsaw there for drying and weighing. Canopy foliage chemistry. So NEON quantifies changes in foliar chemical and structural properties over space and time, as these are commonly associated with key ecological processes, including productivity, decomposition, and herbivory. Similarly, NEON measures the isotopic composition of leaves as well as leaf litter, roots, and soils, thus enabling end users to follow spatio-temporal changes in ecosystem carbon and nitrogen cycles. Plant carbon and nutrient data are generated in collaboration with the Airborne Observation Platform, which is largely responsible for mapping plant chemical and physical characteristics across the observatory using hyperspectral and LiDAR measurements. Our ground-based foliar data will be used to ground truth and validate aerial observation platform measurements. Such data can help the ecological research community refine algorithms to map canopy constituents using hyperspectral data. Additionally, foliar data informs species and site level estimates of canopy chemical constituents and how those change over time. Foliar chemistry data can also provide scientists, managers, and decision makers with important information on ecosystem nutrient status. Comparing these data with those from other ecosystem components, including atmospheric deposition, soils, leaf litter, and surface water, allows investigators to evaluate material fluxes across the landscape. So our sampling method for this protocol involves collecting samples from each species found in our site level sunlit canopy with replicates for common species. We also take digital crown polygons for our woody individuals sampled for unambiguous geolocation of those samples. We also, if necessary, at some sites, take herbaceous clip strips if we have significant open and non-woody areas. The subsamples that are taken from each individual include chlorophyll samples, leaf mass per area samples, and our bulk sample, which is then ground and split into our carbon nitrogen and lignin, isotope ratio, and archive samples. So we can see some of our sampling methods here. On the left, employing the use of a line launcher, which is basically a tiny potato gun kind of thing. So just air pressure beanbag launcher that pulls up a line for us to tow up a saw blade to cut our sample down, as well as use of just an extendable pole printer for more shorter individuals. On the bottom here, we have an example of one of our chlorophyll samples, which are taken immediately once our sample comes down. We quickly take that chlorophyll sample and we flash freeze it by burying it in our cooler that we have with us of dry ice. And we ship those out very quickly. Then we have our Wiley mill, which we use to grind our samples, our micro splitter for splitting, separating those subsamples. And then we have a couple example images of scans that are taken with our leaf mass per area samples. All right, so now that you know a little bit more about our protocols on plant sampling, we can take a look at some actual plant data from Yellowstone. So here we can see the mean alpha diversity per NLCD class at Yellowstone from 2018 to 2020. The mean alpha diversity per NLCD class is simply the number of unique species seen in each plot averaged for each NLCD class. In this figure, the colored dots represent the average alpha diversity among plots across three kinds of NLCD classes, and the gray dots show the actual number of unique species found in each plot across three NLCD classes. We can see that the shrub scrub over here has the highest mean alpha diversity. Additionally, we see that the number of unique species per plot varies greatly in the shrub scrub habitat compared to other habitats. The gray dots, in which each represent a different plot, vary greatly in the shrub scrub habitat, but don't vary much in the other habitat types, aside from a few outliers. This may be because the shrub scrub habitat experiences edge effects or may have a patchy landscape. But basically you can look at this and ask yourself, what are some other reasons why this might happen? Again, this is only looking at a few years of data, which much bigger questions can be asked and observations made once a more robust data set is available, or when it is combined with data from other sources. Which leads me to move on to some case studies that have been done utilizing the data.
Jarrett, could I just interrupt for a second? Someone asked for a clarification. If you could just please again explain what is meant by a tower plot.
Okay, a tower plot just means, so that first map that Jake was showing, where we have our instrument tower, our tower plots are within the tower airshed. So they're within close proximity to our instruments that are also collecting data. And then our other plots that we would be sampling would be for Flora protocols would be our distributed plots, which are scattered across that whole range between Blacktail, Deer Creek, or Undine Falls and the Tower Junction area. So a little bit more variety and a lot of space in between those without the associated instrument data.
Thank you. Great.
Sure thing. Yeah, so we can move on to look at some case studies here that have been done utilizing NEON data. These studies were not performed by NEON, but just involved the use of our data. So this research group outlined a new approach for analyzing phenology observations across a broad array of ecosystems and species. The objective of their work was to develop a multi-region, multi-species model of the onset of spring, i.e. leaf out, and fall, i.e. leaf coloration, for over 100 plant species within 15 temperature ecoregions of the US. They had a 9-year data set of observations from the National Phenology Network and a 4-year data set from NEON to quantify variation between species and regions in sensitivity to environmental drivers for both spring and fall leaf phenophases. They were able to create a good model for predicting this variation. And we can observe how good that is here in just a second here. But if you look at the bottom left figure, you can see the model predicted values versus the on the ground observed dates of spring leaf out for 128 species in panel A and fall leaf coloration for 112 species in panel B. So panel A there and panel B here. Perfect predictions would fall along the 1-to-1 line, so that line in each of these. Most of the data fall along that 1-to-1 line, black line, showing that the model accurately predicts leaf out and fall leaf coloration, the red being the 95% confidence interval. So here, the figure on the left shows you what the model predicts for the mean sensitives among species and regions of deciduous trees and shrubs phenology to environmental drivers. Panels A, spring leaf out, and panel B, fall leaf coloration, show estimated mean sensitivity, the 95% confidence interval for each environmental driver, plus the baseline hazard rate or intercept. Positive coefficients indicate, so above the line, indicate climate forcing variables that prolong the initial state, i.e. leafless state for spring, green leaf state for fall, and therefore delay the onset of the phenological transition. So B figure, this figure is telling us that the model predicts that spring leaf out from panel A was generally promoted by longer days, spring growing degree day accumulation, overwinter chilling, and was suppressed by frost events, whereas fall leaf color was promoted by shorter days and cold accumulation. The figure on the right shows the sensitivity of spring leaf out to growing degree days for each species. Negative parameter estimates, so below the line there, reflect species where growing degree days accelerate spring leaf out. So we can see this varies by species, but definitely majority of them fall below that, where it is accelerating spring leaf out, having that sensitivity to growing degree days. But overall, this study showed that we can use citizen science and national scale monitoring phenology data to develop accurate models of plant phenology. This is important because these kinds of models are normally limited by the intensive phenological monitoring effort required to generate sufficient data to parameterize each model. And NEON provides an immense amount of this data for free. So this research group here used NEON plant data to explore two famous contradictory hypotheses regarding factors influencing the outcome of biological invasions of non-native species. First, that non-native species closely related to native species would be more likely to successfully establish because they might share adaptations to the local environment. And conversely, second, that lack of competitive exclusion would facilitate the establishment of alien invaders genetically distinct from the native flora. To date, no consensus has been reached regarding these opposing hypotheses. Specifically, this research group used plant data from NEON and the US Forest Service dataset to examine patterns of taxonomic and phylogenetic relatedness between native and non-native taxa across thousands of locations, ranging in size and extent from local to regional scales. They also looked at how temperature and precipitation affect biological invasions. The figure the figure you can see here shows the data that they use from across the US. The blue circles, which are quite plentiful, represent Forest Service plots, and orange diamonds represent neon plots. So here, their results show that the presence of native species closely related to invasive species is more likely to predict invasion success at larger spatial extents and harsher climates. The figure on the left shows you the relationship between the relatedness of invasive species and native species and spatial scale. For box A here, we can see that the expected number of invasive species with the same genus as native species, otherwise called cogeners, in a plot increases with scale. So as scale increases, so does relatedness. Box B shows you the same relationship, but using mean phylogenetic distance instead of species with the same genus. Here we see that as the scale gets to larger, the mean phylogenetic distance decreases. So invasive and native species become more related. Boxes A and B in the figure on the right show that mean phylogenetic distance, so less related, tended to increase with temperature and precipitation at local scales. So we see unrelatedness increasing with increasing temperatures and precipitation. Warmer, wetter environments tend to support more unrelated species and are less harsh, leading to more competition. Whereas at regional scales, boxes C and D, the mean phylogenetic distance decreased in response to increasing climatic variation. So we see relatedness increasing with increasing climatic variation, drier, colder environments that vary more, are more harsh and support less species, leading to less competition. So what does this all mean? They found that hypothesis one, first, that non-native species closely related to native species would be more likely to successfully establish because they might share adaptations to the local environment. This happens at a larger scale and in harsher environments. This is likely the case because coexisting species need similar traits to survive larger, harsher environments. Whereas hypothesis two, which focuses on competition among related species, happens at smaller, more local scales and less harsh environments. This is likely because at smaller local spatial scales, density-dependent mechanisms associated with limiting similarity, such as competition and enemy escape resistance, become more important. As the environment is generally homogeneous at local scales, individuals are more likely to interact directly and compete over the same pool of limited resources, and thus there may be a limit to the similarity of competing species that can coexist. So, some cool examples there showing what can be done with neon data. And with that, I will pass it back to Jake for just a moment to talk about a few more things.
Cool. Yeah, thanks, Jarrett. I just wanted to do a quick plug for our Assignable Assets program, which is a formal request process to book an AOP flight, like we've been mentioning, so get some LiDAR data for yourself. You can also use a mobile deployment platform, an MDP, which is basically a small tower in that middle picture there. We're trying to get external researchers can basically book an MDP and put it wherever they want and collect their own data, comparable to the NEON data. We've also had researchers adding instruments to our towers and other site infrastructure. So like we have people adding instruments to our towers and to our aquatic sites all the time. And then we have basically access to field sites for observational sampling purposes. So we basically can collect samples for external researchers. So it's sort of like booking the NEON project to, get your own data. And then, so that's cool. And you can check that out on neonscience.org. And then next slide, Jarrett. I wanted to do a quick plug for the neonscience.org/data, which is where you can find all this great data that Jarrett's been talking about. And you can explore and download the Neon data. And there are basically code packages and data product user guides, detailed protocols of all of our protocols, and news on product updates and data releases. So neonscience.org/data and neonscience.org is where you can find all this good stuff. And I guess we don't have a ton of time for questions here, but I think that's all we have for the presentation. So, and that's a cool picture from our phenocam at the top of the tower. So in September, early snow. But yeah, I don't know if did we have any questions in the chat? I don't see